the process of DNA being transcribed into RNA, which takes place in the nucleus. Yeah, so first we have helicase sheets. So helicase comes in here and it unzips the DNA strand. Next, we're looking for that TATA box that we talked about yesterday. So here's the TATA box on this upper strand here. And so there's a few other names for the TATA box. It's called the promoter. And so this is also known as the on-off switch. So if this on-off switch was methylated, Devin, do you know if it was methylated, would this on-off switch be on or off, do you think?
Off.
Off, yes. So if it's methylated, it's going to be off. That means it can't be transcribed. But we're going to leave it not methylated, so it's on. So it's going to be transcribed. So we'll just write that this top strand, the one with the gene, is our template strand, or our coding strand. And this bottom strand down here is our non-coding strand, because this is the one that's got our gene that we're looking to transcribe. And because the TATA box is on, there's no methylation, then transcription factors come in, and they're going to bind to that TATA box. So that's like flipping the switch on. RNA polymerase, or primase. So that would come in just like in replication when it does that primer. It's going to do the RNA molecules here that are going to make the mRNA strand. It first finds the start codon, which is always TAC. So that's going to be our start codon in mRNA. So then it would just follow along and keep transcribing this gene. And then there's segments of the gene called introns, which are just portions that are not actually coded for the gene. So they're like the junk portions of the genetic code. So then we get to this last portion, C. This is the stop codon. So in RNA language, it's UGA. So TAC is always the start codon, but there's actually 3 different stop codons. This is just one of them. So now it still has the introns in here, and these sections that actually do code for something are called exons. So all of these segments are exons, and then the junk segments are introns. And so the first step of processing it would be cutting out the introns. So we would splice out these introns with an enzyme. So then the SNRPs would glue these together, and so you can rewrite it without the introns. So now we've got it with just the genetic code for the gene we're transcribing. Now the mRNA is almost ready to leave the nucleus, but we need to protect it first. So we would add a guanine cap on the one end, and this is what, and then on the other end we'd add the poly-A tail. And the poly-A tail is just a long chain of adenine nucleotides, and then the guanine cap is a specialized guanine nucleotide. So now the mRNA strand is protected, all the introns are taken out, and it's just got the code, so it's ready to take this gene out of the nucleus